All right, thank you for joining us this morning for the workshop on Airway Physiology directed therapies. I'm Courtney Reichart at Washington University, and my co-chair is Dan DeVore. And so the session today, we're hoping to talk about non-CFTR-related therapies for improving airway physiology for people with CF. And wanting to look at a broad range of strategies, everything from mucociliary clearance to other ion channels, and also thinking about CF modifier genes that could also be targeted. And in our questions, we're really hoping to get at a discussion of how these therapies could complement CFTR modulators. So hopefully what you take away from the talks then today is that there's a need for non-CFTR related therapies, even in this post-modulator era. An idea of what the recent developments are for non-CFTR related therapies, and also for us to start thinking about new areas that could really benefit from focused study. So we have several presenters this morning that will be covering different aspects of these aims. The first to speak, and I'm sorry about the punctuation, it changed this morning, but is Lauren Ye from Case Western, and she'll be talking about the absence of SLC26A9 function in neonatal mortality in mice. Michael Kim from University of Kansas will be talking about metformin improving mucociliary clearance. Marcus Mall from Berlin will be talking about mucolytics. Sasha Goryanova from University of North Carolina will be talking about ORAI activity. And then Nam Soo Ju will also be talking about mucociliary clearance. And we're really encouraging you to ask lots of questions. We have quite a bit of time for questions in this session. All right, thank you. Okay. All right, so please welcome Lauren Ye, who's going to talk about SLC26A9 function.
Hi, thank you for allowing me to speak today. My name is Lauren Ye. I am a PhD candidate at Case Western Reserve University in Mitch Drum's lab. And today I want to share a little bit of my work validating SLC26A9, a modifier gene candidate, and some of the phenotypic observations that we've seen during the course of this work. I have nothing to disclose. So we first started this work actually in a completely different system. This was from a genome-wide association study for meconium ileus, actually, that was published by Lisa Strugg's group in Toronto in 2019. And this study looked at people who have CF, who either had or didn't have meconium ileus, and identified single nucleotide polymorphisms that associated with those two. And in particular, we were looking at SLC26A9, which is on the first chromosome, as a potential modifier gene candidate. This graphic by Ann Harris's group is a really good representation of modifier genes in general. And I'd like to direct your attention to the bottom wedge where SLC26A9 is. And as you can see, it's associated with meconium ileus, ivacaftor efficacy, as well as CF-related diabetes. But curiously, it really has not been a traditional lung modifier gene candidate. So what does SLC26A9 do? It has a number of functions, depending on where you're looking at it. It could be a chloride or bicarbonate exchanger, a chloride channel, or a chloride and sodium symporter. It does depend on which tissues you're looking at. In humans, the biggest expressor is in the stomach, as well as in the lungs, although it has also been studied in relation to other developmental works, in early hearing and neurological studies, as well as in diabetes. And when we look at the expression in the mouse, and an adult mouse, we do see a high expression in the stomach and the lungs, as you would expect, and lower levels elsewhere. Just as a quick overview, because it's unlikely you're familiar with a lot of the published background, this gene has been studied in a variety of model organisms, and the gene is very homologous. Just a couple of studies that I pulled out. So it looked at NCBI variants and saw different activity and in different proteins in various variants. There have been a number of works looking at the combination of CFTR and SLC26A9 in terms of the interaction or potential co-regulation. And there's HBE data that CFTR levels increase in the presence of SLC26A9. And finally, there have been suggestions that it could play a role as mucociliary clearance in drawing in ions and helping to hydrate that layer. But I'd also like to point out that the former studies that have been done in mice have been done in a mixed background. And as you may know, mixed background mice are hardier, and it makes it a little more complicated for the genetic analysis when you're not on a single background. One final study that I'd like to point out is one followed up by Lisa Strug's group, just published this year. And this was a bioinformatic paper looking at SLC26A9 as a therapeutic lung target. In this paper, they investigated SLC26A9 function in people who had CF, pre- and post-modulators in the general population, as well as those in COPD, a different lung disorder. In this paper, they associated a particular genotype with greater lung function in CF individuals without approved therapies. And this showed evidence that SLC26A9 may in fact be a good therapeutic target for other lung airway obstructive disorders like COPD. But since I'm from Cleveland and we're part of the CF Mouse Resource Center and everything's on the Black 6 background, we really wanted to have a common background for this modifier gene candidate as well. But we had a really hard time getting live animals mutant animals to survive. And through this graphic here, you can see that it actually was a little bit more severe than we'd initially thought. We were getting less than 1 in 100 surviving to weaning, and in fact, even just past day 1. However, if you looked at the ratio of animal genotypes just before birth, they were in perfect normal Mendelian ratios. And if you looked at these animals on the day of birth, they seem to be having difficulty breathing. Anecdotally, they seem to be gasping for breath, as well as having a blue discoloration suggesting deoxygenation. For anybody who's not familiar with mouse biology, the C57 Black 6 has a 19-day gestation period, followed by day 0, which was birth. Normally, we would genotype at around day 5 or 6, and then that would be followed by weaning on day 21, and then the rest of the adult life separate. So our experimental design was to take the litter at day 0 and take them for mouse MRI, and then in parallel to go to histology as well as do genotyping. So why MRI? Well, this actually is a very simple principle, the typical school example of a water glass with a pencil in it, where the pencil is still straight, but because of the changes of wavelength in our perception as it goes through different medium, it appears to be bent to us. And this work was, this principle was, the proof of principle was validated by Chris Flask in the Imaging Research Corps at Case Western, as well as his research associates, Bernadette Urokwu. And he really did the fundamental proof of concept work with this by filling lungs and then being able to show how MRI was able to detect the differences. So our hypothesis is that if we looked at a normal wild-type animal who had air-filled lungs, that the magnetic field passing through the fluid-filled body going into the air-filled lungs would then have a distortion that would be detectable as black space on the MRI. But if, however, we were looking at a mutant animal, such as those with the blue lungs, that magnetic field would pass through the fluid-filled body into the fluid-filled lungs and then be detectable as a gray area on MRI. And in fact, that's exactly what we saw. So if you see on the left here in the red box, This is a representative animal who is a wild type for SLC26A9. And you can see that that lung area is filled with that black space similar to the background. But on the right, the SLC26A9 mutant animal, that lung space is cloudy, as you can see in the blue box. And to quantify this, we looked at pixelation scores for the surrounding muscle as well as the lung space that was divided in a ratio and then graphed in this chart here, where you can see that the wild type in red have a lower ratio pixelation score, as you would expect from a larger denominator, than the A9s do. We also looked at histological differences, and this is preliminary work, so we'd love to talk about it. But looking at these arrows, you can see on the right side, the SLC26A9 mutant animal seems to have a different morphology right around the bronchioles that might suggest edema. That is not seen on the left side for the SLC26A9 wild type animals. Additionally, there seems to be more penetrance of blood in the wild type animal that we're not seeing quite as much in the wild type, or the SLC26A9 mutant. But this work is very preliminary, so we still have more to do on this. We then wanted to look at where SLC26A9 was located in tissues, and we started by using an immunohistochemistry protein antibody. However, although this antibody worked really well in the stomach, as you can see in the top two panels, the antibody really did not work well in the lung at all, and it was very difficult for us to detect it above background. So we turned to a different strategy using RNAscope. And from literature, we would expect that the SLC26A9 would be expressed in alveolar type 2 cells, which are the major surfactant secreting cells of the body. And to look at this, we looked at probes for SLC26A9, CFTR, as well as surfactant protein C, which is that major marker of alveolar type 2 cells. And what you can see here is that there's really robust co-localization between the surfactant marker in green, SLC26A9 in red, as well as CFTR. So in summary, SLC26A9 should be considered a lung modifier gene. SLC26A9 is expressed in alveolar type 2 cells and may play a role in surfactant secretion control. And MRI allows us to visualize abnormal fluid in the lungs. In mice, this does appear to be background dependent, suggesting that other genes play a role in its function. And so for next steps, we would like to further investigate the combination with cystic fibrosis mutations. On those MRI images from before, we actually had a variety of CFTR genotypes. So it really seems to be more driven by SLC26A9 on this particular background. However, as you can see from the middle panel, the survival of SLC26A9 animals really seems to be background dependent. So a mixed background, even in our hands, gives a normal 1 to 2 to 1 ratio of survival. But on the black 6 background, it's a 1 to 2 ratio with no mutant animals surviving. So this allows us an opportunity to see what differential genes are causing different survival rates on different genetic backgrounds. And so we're currently doing directed breeding strategies to try to narrow down those regions that confer the survival advantage. And this will allow us to explore possible new therapeutic targets as well as gene interactions. The significance for cystic fibrosis is that SLC26A9 may still yet provide another tool in the pulmonary arsenal. It may represent a possible therapeutic target that is independent of CFTR functionality or expression levels, as we're seeing both in our mice as well as in the COPD research done by Lisa Strug. And it may be another gene to consider for personalized medicine. And I'd like to thank my lab, who's been instrumental in helping with all of this, the Mouse Resource Center, as well as Chris Flask and his team. And I am happy to take any questions.
If I could ask the first question, do Do you have any, so you mentioned that SLIC-26A9 has been shown to conduct potentially multiple different ions. Do you have any idea which those properties might be influential in the type 2 cells that you're seeing it in?
I think that there's been studies shown that there's chloride transport, definitely. I'm not entirely sure which one is currently expressed in there. A lot of the studies that have been done previously showing the ion transport functions have been done outside of tissues in cellular models. And so a lot of times, if you're looking at what can be passed through, it may be different from what actually is being passed through. So I'll need further work to look at that.
We have a question in the app. Is this on? Are there any known drugs that already target SLC?
I actually saw a poster for a specific SLC26A9 inhibitor that looks promising. So I don't remember the poster number, but I direct your attention there. That looked very promising.
Do you have any plans to look at the impact on the intestines? Sorry. Is it on now? Okay. Do you have any plans to look at the impact of the mutation in the intestines?
Yes, actually, so my PhD research is actually focused on intestinal research, looking at meconium modifier genes. And so once we get past the survival issue, we would like to return back to looking at the intestinal effect of SLC26A9 in the intestines.
Are there any audience questions?
I'm sorry. No, the heterozygous, so the question was if we have the mutants that don't survive, does heterozygous have the same phenotype? And the heterozygous actually do survive just fine, so it requires both copies to be knocked out to have function.
Nice presentation, very nice imaging data. So regarding the conclusion that they have more fluid in the lung, have you also done weight to dry measurements over time to see if they have a problem with fluid clearance?
So we,
With the co-expression with the surfactant, it may also be that there is a problem in surfactant homeostasis and expansion of the LPD that may potentially lead also to a similar change in the image.
So the question was, have we looked at our imaging with the wet to dry and looked at the clearance rate of surfactant in these animals? And so the answer is no, we haven't looked at anything looking at a durational timeline. So the measure just at the one point for now, but we have thought about doing something to show a rate of clearance.
So in the introduction, you said that SLC26A9 is a modifier of CF. And now the problem here is that you're having a neonatal mortality in the past. So if it is a modifier of CF, can you utilize the drugs? I do not know in which direction it is modifying. Can you use the correctors potentiators in the mom, like a pregnant mom, and have this survival of the pups, or use inhibitors of CFTR in the other direction, and then you can get rid of this mortality? Just, I don't know.
So the question was, can we use CFTR modulators or inhibitors in the pregnant mom to facilitate the survival of the mutant animals? And what I would say for this is that because we're seeing a survival impact in mixed background animals, that it's probably a secondary gene that's affecting and modifying SLC26A9 as opposed to being directly a CFTR-related problem. So we haven't done anything like that at this point, but it's a good thought. Thank you.
So I'm not that good with SLC26A9 or with lungs. But one of the things that it looked like was, and you said it was in type 2 cells that do surfactant, one of the things that jumped out at me, I didn't see much in the way of inflation in the lung histology that you showed. And so are you in fact getting much inflation at all in any of your alveoli in the animals and you're suffocating them down? Because the amount of fluid in the lung didn't strike me as being all that huge.
Mm-hmm.
But the total absence of air and inflation was just, so surprising.
Mm-hmm.
So are you getting any inflation at all?
So I'm not totally sure. We'll have to finish quantifying that by histology to see if there is a significant difference between the two tissues. What I can say is that with the MRI, the signal, it actually degrades quite a bit. So the fact that we were seeing that much cloudiness already was fairly significant in terms of fluid in the lungs. But we'll still have to do further work on this.
But what you're seeing with the MRI is the absence of air. You're not necessarily seeing fluid. Is that correct?
Yes.
There's one more question in the chat. Can you comment on candidate genes that revert the effects of the SLC mutations when you outcross the mice?
So we're still working on this, so I don't have any candidates off the top of my hand to give you, but we'll look forward to updating you on this at a later date. Thank you.
All right, thank you for all of the discussion, and let's thank Lauren. All right, please welcome the next speaker, Michael Kim.
Can I use a laser pointer? Is it working or?
This one works.
Oh, it's not a good angle. Yeah. Okay. All right. Okay. Good morning. I'd first just like to thank the organizers for giving me the opportunity to speak and to talk about our work this morning. Okay. I have no disclosures. Okay. So it's been fairly well established that impaired glucose tolerance is correlated with lung function decline in CF. And so in this 2000 study by Carlos Mia and colleagues, they showed that those with CF-related diabetes, or CFRD, had a greater rate of lung function decline, both FEV1 and FVC, compared to those with CF with normal glucose tolerance, and even with impaired glucose tolerance. But it's important to note that these studies were conducted before the introduction of highly effective modulator therapy. And as we know, highly effective modulators have really led to dramatic improvements in lung function in most people with CF who are eligible for these therapies. However, it's unclear the impact these modulators have on lung function decline in CFRD. And so our recent study shed some insight into this question. Basically, we found that CFRD is associated with worse lung function trajectory despite modulator use, and in this case, ivacaftor. And so these are data analyzed from the CF Foundation patient registry, and we found that both females and males with CFRD on ivacaftor showed a worse lung function or ppFEV1 trajectory compared to those who did not have a diagnosis of CFRD. Whoops, sorry. Okay. Unfortunately, the prevalence of CFRD increases over time, and even with modulators, there still seems to be a trend to increase. And so, unfortunately, even those on modulators are likely to have a prevalence of CFRD. There's a higher prevalence of CFRD with age. So it doesn't seem like CFRD will be around for a while, basically, is the point. But it's not just CFRD, it's also hyperglycemic excursions that seem to correlate with lung function decline. And so in this 2018 study by Christine Chan, they used continuous glucose monitoring to basically measure glucose levels in people with CFRD. Sorry, it's distracting. And so what they found was that the frequency of hyperglycemic excursions, in this case defined as glucose greater than 200 milligrams per deciliter, the frequency of hyperglycemic excursions were correlated with lung function decline, in this case a negative slope of FEV1. And so our group really is interested in really trying to figure out how hyperglycemia causes lung function decline in CF. And so we know that CF and CFRD affect multiple organ systems, leading to poor outcomes. But we're particularly interested in how elevated glucose affects mucociliary function. So I don't think I need to, I won't have time to really explain how CFTR dysfunction leads to impaired mucociliary clearance. I'm pretty sure you're all very aware of the mechanism, so I'm not going to dwell too much on that here. But I do want to emphasize the importance of other ion channels that on their own are important for airway hydration. So for example, activation of TMEM16A, or calcium-activated chloride channels, have been shown to be important for airway hydration as a kind of compensatory mechanism for the lack of CFTR function. And our group has shown the importance of the so-called BK potassium channel, which is also important for promoting chloride secretion, but on its own is important for mucus hydration. And so this work was shown by Matthias Salate and his group. The BK channel can basically be detected in oozing chambers in air-liquid interface cultures of primary human airway epithelial cells by stimulation with ATP. The BK channel is calcium activated. And so what you see on the left basically is ATP stimulation in oozing chambers. This leads to opening of the channel and essentially potassium efflux and a net negative change in short circuit conductance. And so the key experiment is that when you knock down the major pore-forming subunit of the BK channel, this not only leads to a significant reduction in BK function, but if you look on the right, it basically abolishes ciliary beat frequency. And so this is in normal cells, so there's functional CFTR and functional CACC. So this demonstrates the importance of the BK channel in maintaining mucociliary clearance. So in our previous study we published last year, or 2 years ago, we found that chronic hyperglycemia causes BK dysfunction and subsequent ASL depletion in primary CF bronchial epithelial cells in vitro. And so for these experiments, we cultured primary CF cells at the air-liquid interface, and we cultured them under either normal glucose conditions, which we defined as 5.5 millimolar glucose, which is essentially equivalent to 100 milligrams per deciliter, or we cultured them under high glucose conditions, that being 12.5 millimolar glucose, or the equivalent of 225 milligrams per deciliter. And basically what we found is that CF cells that were grown under conditions of chronic hyperglycemia had a significant decrease in BK activity and subsequent decrease in airway surface liquid volumes. Now we're also interested in how short-term elevations in glucose potentially affect ion channel function to kind of more closely mirror these hyperglycemic excursions that are seen in people with CF. And so for these experiments, we cultured primary CF cells under conditions of normal glucose, but then just switched them for 24 hours under conditions of high glucose. And similar to what we saw with chronic hyperglycemia, if you switch these cells to high glucose, you see a significant reduction in BK function, leading to reduction in ASL volumes. And I just want to emphasize, I forgot to mention, that this is in the absence of any kind of modulators, so there's basically no CFTR function in these experiments. So we're interested in identifying therapeutic approaches to kind of counter these effects of high glucose on ion channel function, and one of the therapies that we've pursued is metformin. And so Most of you are probably familiar with metformin. It's commonly prescribed for type 2 diabetes. I think there's over 90 million prescriptions in the US alone. The interesting thing about metformin is that independent of its glucose-lowering abilities, it also has anti-inflammatory properties and is actually used off-label to treat a number of different diseases. Now, the only caveat to the use of metformin is that high concentrations of metformin have been shown to inhibit CFTR and ENaC conductance. And so we basically pursued a strategy using low and clinically relevant doses of metformin. And so for these subsequent experiments, we're using it at 1 micromolar. And so under the previous paradigm, we basically found that when you treat these cells that are challenged with high glucose with metformin, you can significantly increase BK conductance as well as ASL volumes. And the important thing to note is that this is the absence of any effects on glucose. And so these are glucose levels in the media. Now, I'm sure people are wondering what happens to CFTR function, in this case, modular corrected CFTR function. And so under the similar paradigm, we cultured homozygous F508del CFB cells under conditions of normal glucose and then switched them, again, for 24 hours with high glucose. But in this case, they were treated with a triple combination of elexacaftor, tezacaftor, and ivacaftor, or ETI. And basically what we found is that whether they were cultured throughout in normal glucose or switched to high glucose, it seems that the glucose did not affect the CFTR function. However, what was interesting was that cells challenged with high glucose, although ETI caused a significant increase in modulator-corrected CFTR function, it did not lead to a significant increase or significant improvement in ASL volumes. However, when we added 1 micromolar metformin, we could actually further enhance CFTR function in cells that were challenged with high glucose, and this in effect led to a significant improvement in ASL volumes. So what we found was that the increase in CFTR function correlated with an increase in CFTR mRNA. And we also looked at secreted HMGB1, or high mobility group box protein 1. HMGB1 is a so-called damage-associated molecular pattern that's increased under conditions of cellular stress like hyperglycemia. And what we found was that metformin was able to effectively reduce the concentrations of secreted HMGB1. And so a little bit more about HMGB1, like I said, it functions as a DAM, but its normal function is actually as a DNA binding protein. It's important for transcription. But under conditions of cellular stress, it will actually translocate out of the nucleus into the cytosol and become secreted. And once it's secreted, it can bind to these so-called pattern recognition receptors like TLR2, TLR4, or RAGE. And so it can bind to these receptors to mediate a pro-inflammatory cascade. HMGB1 levels have been known to be elevated in CF, and actually particularly at the onset of CF-related diabetes. And so in this study, they found that serum levels of HMGB1, in this case greater than 9.5 nanograms per mil, as well as sputum levels of HMGB1, in this case greater than 5.3 nanograms per mil, were associated with basically worse lung function trajectory. And so we were interested in trying to understand if HMGB1 could be possibly mediating some of these effects of high glucose. And so for these experiments, these were homozygous F508del CFB cells, again cultured under normal glucose conditions. And in this case, they were all treated with ETI for 24 hours. However, when we added HMGB1 basolaterally at physiologically relevant concentrations, in this case we used 10 nanograms per mil, we saw a significant decrease in ASL volumes, as well as ciliary beating, despite the presence of ETI. But if we added back metformin, in this case 1 micromolar again for 24 hours, the metformin could completely reverse the effects of HMGB1 on ASL volumes and ciliary beating. However, what we found was HMGB1 did not seem to impact ETI-corrected CFTR function, as you can see here, and in effect, metformin also didn't further improve CFTR function. We looked at HMGB1 actually in higher doses, and even at higher doses, we didn't see an effect on ETI-corrected CFTR conductance. Previous studies have shown that high concentrations of HMGB1 can actually increase ENaC, and this could potentially explain the ASL depletion phenotype that we see. However, we didn't see an increase in ENaC or a change in CACC as a result of treatment with HMGB1. However, what we did find was that HMGB1 after 24 hours led to a significant decrease in BK channel function, similar to what we see with high glucose. And in this case, the addition of metformin was again able to improve or rescue HMGB1-induced BK dysfunction. We also found that HMGB1 caused inflammation, which is not surprising. We looked at expression of IL-1 beta, TNF-alpha, TGF-beta1 mRNAs, as well as TGF-beta1 protein. HMGB1 caused an increase in all of these cytokines, and metformin was able to significantly reduce expression of all of these cytokines as well. And so, as I mentioned previously, HMGB1 can act through multiple receptors and through multiple pathways. And so we're trying to now understand which pathways are responsible for mediating some of these effects. And so in preliminary experiments, we've targeted the TLR4 and RAGE signaling axis using inhibitors. In this case, TAC242 is a TLR4 inhibitor or antagonist. S-RAGE is soluble RAGE. This is a decoy receptor for RAGE. And by blocking both TLR4 and RAGE, or TLR4 and either RAGE, we see improvements in ASL volumes, suggesting that HMGB1 may be signaling through this axis to mediate its effects. So lastly, our in vitro data with metformin in terms of being able to improve ion channel function, mucociliary function, led us to propose a clinical trial of metformin in those with CFRD on modulator therapy. So this study began this year and is currently enrolling, and we hope to be able to present the data at future meetings. So in conclusion, we showed that high glucose, both chronic and acute, or not acute, but short-term elevations cause BK dysfunction and ASL depletion in the absence of modulators in primary CF cells. Turns out metformin does a really good job in improving CFTR function as well as BK function and ASL volumes in vitro in ETI-treated cells. But we found that HMGB1, which again is elevated in CFRD, can cause depletion of ASL as well as reductions in CBF. And we suspect that it's due to BK dysfunction, but that metformin can actually reverse this as well. And finally, metformin reduces the inflammation that's induced by HMGB1 in these cells. And so lastly, I just want to thank the team. This is the team at KU Medical Center led by Dr. Matthias Salate. These are the donor agencies, and of course we want to thank the people with the CF that donated their explanted lungs. And of course the funding through the Cystic Fibrosis Foundation and NIH that makes this work possible. Thank you. Any questions?
So if I can ask, I believe your group has shown, right, that these cytokines decrease BK function by affecting the gamma subunit?
Yeah.
And so do you know if that's true of high glucose as well, or the HMGB1?
So under conditions of chronic hyperglycemia, we did see a reduction. So the question is, the gamma subunit of BK is important for its function in airway epithelial cells, and reduced expression of LRC26 by TGF-beta, for example, we suspect is responsible for the reduction in BK function. And under conditions of chronic hyperglycemia, we did see a reduction in LRC26, which we suspect is causing the decrease in BK conductance. I didn't address that here, though.
That's fine. So would you also then propose that potentially BK potentiators would be a useful therapeutic?
Yes, and we've explored that idea, but there's a few, but I don't know how effective they are, or sorry, I shouldn't say that, in terms of going into clinical studies, right? I mean, there's none that are approved. And so this is why we kind of focused on therapeutics that are FDA approved, that we can kind of get right to to the clinic. And metformin actually does a spectacular job of improving BK function under conditions of hyperglycemia.
We have a question in the chat, which is, can you discuss what outcomes will be measured in your clinical study, i.e. sputum for cytokines or microbiology or just FEV1?
So, for the clinical study, so we have the primary outcome is basically improvement in ion channel function, and we are right now doing nasal potential difference measurements, to look at improvement in both CFTR as well as BK function, and also doing nasal samples for expression of cytokines and LRC26 actually as well.
So you chose to use physiologically relevant glucose elevation for your high glucose levels. Did you also see a rise in the ASL glucose levels equally?
I'm sorry, I see a rise in the ASL.
Glucose levels, glucose concentration.
Oh, in terms of measuring glucose in the ASL. Actually, we have not done that. He was asking if we measured the glucose levels in the ASL. And no, we haven't done that. That's a good idea.
I guess one last question is how, so if you start disentangling the anti-inflammatory effects of metformin from the other effects, I guess my question is how does like another type of anti-inflammatory compare in being able to have a similar effect? So like ibuprofen even or something.
So in terms of mechanism, I think metformin has multiple effects. I mean, it is an AMP kinase activator. But it also counters rage signaling, and so I didn't really explore that and talk about that much. And there's some studies that suggest that metformin can also bind to HMGB1 itself. And so in terms of exact mechanism, we're exploring that, yes.
All right, any final questions from the audience? All right, thank you.
Thank you.
All right. Please welcome the next speaker, Marcus Small.
Yeah, thank you for the opportunity to present our results on a new mucolytic, MUC31, as a potential new therapeutic for cystic fibrosis. And I'm presenting this data on behalf of Annalisa Ardante, who is the first author of this work and unfortunately cannot be here today. And I'm trying to advance. Use this one. Click on that now.
Just bring the mouse down.
Okay. Okay. No. Yes, okay. So here are my disclosures related to the development of new therapeutics for cystic fibrosis. And here is the problem, the mucus plugging that we are very well familiar with in CF, but that is also an emerging topic of relevance in other chronic lung diseases, including COPD and severe asthma, which obviously causes airflow obstruction, but also is a chronic nidus for chronic infection, trigger for chronic inflammation, which is really the key issue in progressive remodeling and damage of the lung. And so far, we do not really have effective therapeutics targeting this problem in the lung. So in addition to airway surface liquid depletion due to the ion transport defect, there's an emerging theme that increased cross-linking of mucin polymers due to oxidation due to reactive oxygen species that are released in chronic airway inflammation, contribute to increased elasticity of the CF mucus. And this has been shown here, for example, in a previous work by John Fahy's group, where they showed that when they oxidize mucus, that this leads predominantly to an increase in the elastic mucus, as well as the viscous modulus of mucus in rheology studies. And in this work, they also found that a thiol-modified sugar, monosaccharide, galactose, has potential mucolytic activity as a reducing agent, with a free thiol attacking these excessive disulfide bonds, as shown here, where they also found that it is more potent than the only approved reducing agent we have in the clinic so far, N-acetyl-cysteine. Now, based on these results, they have, the group of John Fahy has designed and synthesized a novel thiosaccharide, which is called MUC31. And the aim of this study was really to do a preclinical evaluation of this molecule, both by rheological measurements in CF sputum, as you can see on the left, where we compared efficacy of MUC31 with pulmozyme and NAC on the viscoelastic properties of CF sputum, but then also preclinical testing in the beta-ENaC mouse as a model with CF-like lung disease, where we treated both wild-type and beta-ENaC transgenic mice with a compound or vehicle, and then looked at the mucus content in the airways by histology and inflammatory cell counts in BAL, basically in 3 settings, as you will see later, in an acute treatment paradigm where they were treated pretty intensely for 1 day, but then also chronic treatment in adult mice with established lung disease and preventive treatment in neonatal mice. Now, first, the data on the sputum rheology. As you can see here in the comparison of MUC31 and pulmozyme, you can see on the left that MUC31 was faster in bringing the elastic modulus, or reducing the elastic modulus to 50% of its initial value than pulmozyme. And on the right, you can see that a larger percentage of the sputum samples treated with MUC31 reduced the elastic modulus by at least or greater than 50%. Now, on the next slide, you can see the comparison with NAC. And again, MUC31 was faster in reducing the elastic modulus to 50% of the initial value. And again, also larger percentage of the CF sputum samples were shown to reduce the elastic modulus greater than 50% over time, supporting that this molecule is a more effective mucolytic, at least in patient sputum, than either pulmozyme or NAC. And this in vitro data in the sputum then really led to the next step of in vivo testing in the beta-ENaC transgenic mouse, where, as I already mentioned, we started with an acute, testing the acute effect, and this means that adult beta-ENaC transgenic mice that already had established mucus plugs were treated 3 times in 1 day, and then after another 2 hours, the endpoint studies were performed. And as you can see in the histology and the quantification, that both the total mucus, or the total airway mucus, was significantly reduced. We then also separated the intraluminal mucus from the intraepithelial mucus, which reflects goblet cell metaplasia. And there you can see that this effect was mainly relied on the reduction of the intraluminal mucus. And interestingly, even with this short therapy, we also saw a reduction in the total inflammatory cells in this setting. Now, in the next study, in adult mice that were now treated twice daily for a period of 2 weeks, you can see that we also found a reduction in the total and intraluminal mucus, and also a reduction in the total inflammatory cells, which was mainly attributed to macrophages. Also a trend towards reduced chronic airway neutrophilia. And this reduction in airway macrophages was also associated with a significant reduction in TNF-alpha that probably predominantly comes from the activated macrophages in the airways. So then, as a next step, we went to ask the question if this strategy can also be used as a preventive therapy. And to do this, we treated mice again twice daily, actually from birth, from the first day of life, again for a period of 2 weeks. And the beta-ENaC transgenic mouse on this background has a spontaneous mortality of about 30 to 40%. And as you can see on the right upper panel, the treatment reduced this spontaneous mortality that is caused by early airway mucus plugging significantly. And then when we looked at the airway, the path staining and morphometry, you can also see again significant reduction of the mucus plugging. Now here, interestingly, also of not only of the intraluminal mucus, but also of the goblet cell metaplasia. And I will come to that again in my next slide. In this study, we also did mucin western blots using a MUC5B antibody at the bottom, and you can see that especially the high molecular bands, so the highly cross-linked mucins, were also significantly reduced in the animals that were treated with MUC31. We also looked at the effects of inflammation in this setting, and we again found a reduction of macrophages as well as eosinophils. And I do want to mention that there is an age-dependent inflammatory phenotype in this model where juvenile mice have type 2 inflammation with eosinophils as well as IL-13. As you can see on the bottom, in both components of this type 2 inflammation, eosinophils as well as IL-13 were reduced when mucus transport and clearance was improved with this molecule. And I think this reduction in the IL-13 is also the explanation for the reduced goblet cell metaplasia in this age group I showed you on the previous slide. So taken together, we think that this data supports that this new compound has complementary mechanisms of action of mucolysis to pulmozyme by more directly targeting the abnormal structure in terms of excessive disulfide bonds in the mucus gel. And so therefore, we conclude that first, the rheology studies in CF sputum support that this novel thiol-based reducing agent is more potent mucolytic than pulmozyme and NAC, that our preclinical studies in the beta-ENaC transgenic mouse demonstrate that treatment with this compound decreases mucus plugging in vivo, and that this reduces airway inflammation and improves survival of the mice, and that the data support that cleavage of excessive disulfide bonds in mucin polymers is an effective mucolytic strategy, and that the data, especially the in vivo data, also provide a rationale for further clinical development of this compound, both for CF, but potentially also for other muco-obstructive lung diseases. With this, I would like to stop and thank my team in Berlin, especially Annalisa, who really did most of the work and pulled it together, but also John and his team in San Francisco, and his colleagues in Dublin, who designed and synthesized the molecules, and the funding agency for funding. And I thank you for your attention. Be happy to take questions.
I actually have two questions. So one is just like a small technical question, and that's on the Western blot of the mucins. Was that a non-reducing gel then that you ran there, or because seeing the higher molecular weight mucins that were presumably oxidatively cross-linked?
Yes, so this is a technique of an agarose gel that has been developed by Alessandra Livraci in the audience. And used for the first time in the beta-ENaC transgenic mouse, and we strictly followed her protocol, and she can maybe provide all the details in the.
Right.
It's actually or reducing the disease, or if there is some other effect.
Mm-hmm.
Your mucility is a sugar, right?
Yes, and it with a thiol.
With a thiol.
Yes, so it reduces the disulfide bonds.
So
So
if there is the people balanced osmotically, is it possible that it doesn't?
well, that should not play a role. So the vehicle was corrected for potential osmotic, so it has the same osmolarity in the in vivo studies, yeah? And this should also not affect the effect seen on the BAL Western blots.
Yeah, but if you run it,
One question back here.
I could not understand.
What was the motivation for choosing the carbohydrate that you did rather than some of the other ones that are on the mucin chain?
Yes, so I mean, this goes back to the work of John Fahey, and where they started with galactose as a monosaccharide and then went through different preclinical in vitro optimization assays. I think a consideration there also is more around potential pharmacokinetics and pharmacodynamics for further development of this as an inhaled therapeutic.
So, excuse my ignorance of this subject, but I'm curious with the highly effective modulator therapies, is there a difference in the disulfide linkage in the mucus in those patients on those compounds, and how that might impact the MUC31?
That's a very good question, Dan. I mean, so you could also ask, are reducing agents still needed for CF patients on highly effective modulator therapy? So I don't have it here, but we have started looking at rheology in patients at baseline and on ETI. And I mean, this is still sort of, work in progress, but what we see is an improvement of the elastic modulus, but not back to normal, so substantially still abnormal viscoelastic properties that, I think would speak to an additional need of mucolytic agents over time. And then I think there's a real opportunity also for other lung diseases outside CF.
But asking about the delivery in the nasal instillation, measure what your actual delivery
The lung?
to the lung for the airway was, and then the follow-up is that sort of concentration or a minimally concentration feasible for
Yeah, so I think I didn't mention that. So we did not use intranasal, we used intratracheal, at least in the older animals. You're correct, we did the intranasal in the pups. And we did deposition studies before where we showed that we can deposit at least 10% of the compound. And that is a dose that is expected also to be deliverable in patients. Yes. Yeah, so that is a very good question and a very general question or potential problem with any reducing agent. And as you say, other proteins, transmembrane proteins, secreted proteins, could be affected in their structure. The CF airway has a lot of mucus on the surface. Yeah, so one concept would be that it can act on this mucus, and the mucus can then become more transportable and clearable, also clearing and using up the aggressive thiol before it can attack other targets. And so I think, obviously, if you use something like DTT, that will probably kill the lung. So you cannot be too aggressive with this approach. But I think there may be an opportunity at a dosing range where it can have primarily therapeutic effects without having these type of adverse off-target effects.
A couple of questions, one biochemical and one more physiological. So on the biochemical level, with the compound you're going to be breaking down disulfide bonds, and we know there are four intermolecular and intramolecular disulfide bonds. So did you look at that? And second, do you have cumulative effects with molecules that would chelate, for example, divalent cations that are also promoting UC interaction by providing heterostatic bonds?
Yeah, so that's again two good questions. So we have not looked at the specificity of which disulfide bonds are targeted, because you could argue there's physiological, disulfide bonds are important in the physiological folding and the assembly of the network. And the second question is, how important are disulfide bonds versus non-covalent bonds? And could that be another therapeutic target? And we have also not looked at this, but I think there is at least one published compound targeting non-covalent interactions.
So the physiological part now, in your early treatment, do you end up seeing a change in goblet cell hyperplasia that you see in that mouse model as well? So there might be some feedback regulation of nuclear production. Do you see any change?
Yes, exactly. So that's in the juvenile study, there we also saw a reduction in the goblet cell metaplasia. So the question is, how can that be explained? That could be explained potentially by if you have less plugging in the lumen. The goblet cells can make a better job in secreting. But what we also found via this anti-inflammatory effect, that we have less IL-13, as you know, is a very strong driver of goblet cell metaplasia. So I think there are these two alternative, potential explanations for this. But I think this part could be very interesting for type 2 inflammatory diseases such as asthma.
Right. There are a couple questions still in the chat, and if you're okay taking them, we have time, I think. So one is, are there any potential safety concerns or off-target effects of MUC31?
Yes, so as we discussed and brought it up before, other proteins have disulfide bonds that are important for their function. So if they are targeted, that could cause adverse events. But again, if you think about how much stagnant mucus and adhesive mucus there is in CF airways, I think there is a chance that that sort of basically protects from the off-target effects. But that has to be studied, tested, obviously, in clinical trials.
Right. And a follow-up question with that is then, if you're providing, say, or the DNase and MUC31, presumably you'd have to treat at separate times with those so that MUC31 doesn't have an effect on DNase, or is that not the case?
I mean, clinically we treat with pulmozyme once a day. Yeah, so it has a relatively long half-time. Yeah, so if you want, and I mean, that's also something that needs to be addressed in clinical trials. If MUC31 alone is sufficient, or if there are additive or potentially even synergistic effects of targeting the excessive DNA versus the cross-linked mucus gel.
Yeah, I guess my question, I don't word quite right. Does MUC31 impact the activity, even just like biochemically, of pulmozyme?
Not that I know of.
Right. Any final questions from the audience? All right, thank you very much.
Thank you.
Are you okay? Yeah. All right, please join me in welcoming Sasha Goryanova.
Hi everyone, I'm a PhD student in the lab of Robert Tarrant at the University of North Carolina at Chapel Hill, and as such, I have no disclosures. So I'm going to be talking about pro-inflammatory ORI1 activity and how it's elevated in CF patients regardless of the fact that they are on these highly effective modulators, and in this case, they are on Trikafta. So, let's see first I want to talk about pulmonary inflammation in CF. I'm sure most of you know about this, but I want to just give a brief refresher so that you know what I'm going to be referring to in these following slides. So, from birth, CF patients have difficulties with clearing infections and the fact that they have this mucociliary clearance defect. So, at first, when you have this mucus obstruction and bronchiostasis, you have an increase in the bacterial infections that are acute and recurrent in these CF patients. Now, of course, the body does still try to clear them. So there is large amounts of immune cell recruitment, which, as they're trying to clear, causes large amounts of inflammation and, unfortunately, lung scarring because, as I mentioned, these bacterial infections are not able to be cleared. So when these neutrophils keep coming and releasing neutrophil elastase, this does cause lung damage that, unfortunately, leads to progressive loss of lung function. And if it isn't able to be dampened down, these patients often have lung failure that leads to transplantation or, very unfortunately, death. Now, I want to talk about a particular protein in the human body that has a relationship with the immune system and the regulation of the immune system in immune cells. And that particular protein is ORI1. ORI1 is a plasma membrane calcium channel and it is ubiquitously expressed in cells throughout the body and is particularly important in immune cells and calcium homeostasis. So I just want to take you through this really quick. So when you have an antigen-presenting cell that activates T-cell receptors, though I would like to point out that GPCRs can also activate this signaling cascade. The signaling cascade causes the activation of the IP3R receptor, which causes calcium to be released from the endoplasmic reticulum stores. Now, when we are doing this in vitro assays or in vivo, we are using an artificial inhibitor of the circle pump inhibitor that causes the release of the calcium stores from the endoplasmic reticulum, and this allows us to mimic this activation cascade in order to be able to look at this experimentally. Now, once the calcium has left the endoplasmic reticulum, STIM1 senses this depletion and translocates to these plasma membrane junctions and oligomerizes with ORI1. Once STIM1 oligomerizes with ORI1, ORI1 molecules come together, proteins come together in order to form these puncta, and this is done in order to potentiate the calcium signal. Now, once calcium enters the cell, it causes the dephosphorylation of NFAT, which then translates to the nucleus and causes the upregulation of target genes that increases the immune response, release of TNF-alpha, and pro-inflammatory cytokine release. Now, again, this is a very important process that is important for the ability of the body to regulate inflammation and clear infection, but if it's overactive, then it could cause issues. So calcium signaling in CF is dysregulated, and my lab is not the first lab to have said this. There have been many previous articles published on calcium dysregulation in cystic fibrosis. For example, this group is the Hanrahan group, which says that increased ORI1 plasma membrane insertion in CF causes increased insertion due to this defect in CFTR. Then you also have the Vatchell et al., which says that correcting CFTR dysfunction can improve calcium dysregulation. This group said that increased cytostolic calcium stores in CF, and this group also talked about dysregulated calcium signaling actually directly affecting CF neutrophils and diminishing their ability to have antimicrobial killing. So here is just a representative trace in a cystic fibrosis cell line. This was published in the Hanrahan paper about how you have dysregulated calcium signaling in CF. So here you have wild-type CF cells, and then you have the delta F508 bronchial epithelial cell line, and you can see that they have upregulated calcium signaling compared to the wild-type group. And in this particular experiment, they did a calcium switch or a calcium add-back experiment in which they depleted the extracellular environment of calcium and then added it back in order to be able to mimic the activation of ORI1 specifically because it is store-operated calcium release. So the general theory here is that in CF, there is a dysregulation of calcium. So you have normal activated CFTR here, and you have a normal number of activated ORI1 particles, and there is a general homeostasis of this immune cell process. However, in CF, where you have misfolded protein or protein that doesn't work appropriately, you have either more or more overactive ORI1 at the plasma membrane, and unfortunately, this leads to inflammation. Now, previous studies have been done in only CFTR cell lines or in primary human cells. But our hypothesis specifically is that ORI1 is upregulated in CF patients' lungs. And the reason that we think this is particularly important is ORI1 is a well-recognized and validated drug target, so we wanted to be able to further explore this in a basic cell biology perspective in order to strengthen our argument that the development of anti-inflammatory therapeutics through ORI1 is a very viable approach. So the very first thing that I did is looked at a single-cell RNA-seq database called LungMap in order to be able to data mine this ORI1 and STIM1 expression just in a normal human lung. And the reason that I did this at first is because, in fact, nobody has truly studied ORI1 and STIM1 in the lung in situ. And if you can look at this UMAP over here, you can see that ORI1 and STIM1 is pretty universally expressed in the lung. This is not particularly surprising, but what I would like to point out here is that first, there is a particular expression of ORI1 and STIM1 in these macrophages, monocytes, dendritic cells, the immune cells that we're really trying to focus on. And also that where ORI1 is located, there is also STIM1, which again makes sense because STIM1 is ORI1's activating molecule. Now, once we had this general idea that we're going on the right path in terms of looking at ORI1, we then wanted to look at it in actual lung tissue sections that we had available. So we got normal CF and asthma lung tissue sections from our Marsico Lung Institute Tissue Procurement Corps run by Dr. Scott Randell, and we used asthma as another inflammatory disease control. And then we had deparaffeinization and antigen retrieval of it done by the Antigen Retrieval Corps. And I did three-pronged approach where I looked at RNA scope, or RNA in situ hybridization, immunostaining for protein expression, and also looked at the super resolution for puncta analysis, which is when I talked about that activation causes ORI1 to agglomerate together. And I did all of this on the Leica SPA confocal microscope. So first, I'm going to talk about the RNA scope gene expression. And ORI1 and STIM1 gene expression is upregulated in some regions of the CF and asthma lung. So in red here, you can see ORI1 RNA, and here on the bottom in yellow, you can see STIM1 mRNA. Here are the immune cells labeled by IC. You have the interstitia, and then you have the epithelia here. And essentially what we found is that, in fact, there is ORI1 and STIM1 expression in all of these regions, but from the mRNA expression alone, you could not definitively say whether there is or is not any upregulation. There were no consistent trends, and here I show the representative graphs based on the immune cells because that is what we are focusing on here. So then I moved on to the ORI1 and STIM1 protein expression to see if, even though there's not any gene expression differences, there might be the translation and protein expression differences. Again, up here in red is ORI1, and down here in yellow is the STIM1, and you have the same things where this is the interstitia, over here is the epithelia, and then you have the immune cells. Here you can definitely see that there is much greater expression of ORI1 in immune cells compared to the epithelia and the interstitia. But despite this observation, which is still very important, there was again no consistent trends or differences in the actual expression between the normal and non-inflamed lungs compared to the cystic fibrosis and asthma. So then we just wanted to think about, well, just because there isn't actual differences in expression doesn't mean that there isn't differences in activation, because a little bit of calcium can in fact go a really long way in being able to propagate any signaling cascades. So I just want to reorient you really quickly to this important thing about punctive formation and how ORI1 and STIM1 have to oligomerize in order for this ORI1 channel to activate. So this is just a similar representation that I showed in the original signaling cascade, where you have STIM1 has oligomerized to ORI1 and caused ORI1 to form these puncta, and this is the indication you have that ORI1 is activated. And for a imaging representation of what this looks like, you have ORI1 YFP and STIM1M cherry co-transfected HEC293 T cells, and you can see that in control, so no treatment done, there is a distinct localization of ORI1 and then STIM1 separately. However, when you add dapsibgargan, that circuit pump inhibitor I had mentioned at the beginning, which causes the calcium to leave the endoplasmic reticulum and ORI1 to activate, you can see that there is very distinct co-localization between ORI1 and STIM1, as well as these punctate formed with ORI1 and STIM1, which indicates that there is very strong amounts of activation. So I just want you to keep this in mind as I'm talking about the findings through the lung tissue sections. So we did in fact find that ORI1 and STIM1 co-localization is significantly increased in CF and asthma lungs. And here on the left, I just want to show you a quick diagram of what this would look like when I'm imaging. So I used the Leica imaging software, and you can see here the ORI1 on the left and the STIM1 on the right, and they highlight the particles in white that are considered to be over a particular threshold, and they show an overlap. So here you have an ORI1 highly expressing region here, a STIM1 highly expressing region on the bottom, and then here in the middle is where the software would consider the overlap of where ORI1 and STIM1 are localized to the same area. So we found that in the epithelia, interstitia, and the immune cells, there is significant co-localization of ORI1 and STIM1 that is not seen in the normal, non-inflamed lungs. And of particular interest, we do not see this in the alveolar cells. And this is particularly important because CF is largely considered an airway disease rather than an alveolar disease, so it was a nice internal control to show that our co-localization is meaningful. So now I want to move on to looking at those puncta, that agglomeration of ORI1 that occurs when ORI1 is activated to potentiate this signal. So I was looking at lung macrophages in those same lung sections you saw for RNA scope and protein staining. And you can see here that you have ORI1 on the top and STIM1 on the bottom, and you can see that in the CF and the asthma lung macrophages, there is greater ORI1 puncta formation than in the normal, which indicates that there is greater activation of this ORI1 channel. Now, I do want to point out that I was unable to compare neutrophils in these lungs because the normal lungs did not have any infiltration of neutrophils due to the lack of an infection or inflammation. However, I do know that CF is largely considered a neutrophil-dominated disease, so I did want to look at neutrophils in CF patients compared to normal. So what I did is I got normal and CF blood samples, and then I did neutrophil isolation on them, immunostaining, and did the same puncta analysis that you just saw occur in the lung tissue sections in order to be able to compare the CF and normal neutrophils. So ORI1 puncta are significantly larger in CF blood neutrophils, and here are the demographics of the group. It was a relatively small cohort, but I do want to draw your attention to the fact that all of these patients are on Trikafta. And despite the fact that all of these patients are on Trikafta, they still have a relatively low lung function, which indicates that they do still have persistent lung disease and that they do still have chronic inflammation. And just to reiterate that Trikafta is not a cure, and there still needs to be more therapies designed for the individuals who do not fully respond to Trikafta, or the ones that are not able to respond to it at all. So when you look here at the neutrophils, I want you to direct your attention to the top row first, where you have the normal neutrophils. So you have this staining for ORI1 in the vehicle, and then when you add thapsigargan, that circle pump inhibitor that activates ORI1, you can see that there has been puncta formation occurring, indicating that this thapsigargan activated the neutrophils. Now, when you look at the bottom row and you see the CF neutrophils, you can see that thapsigargan did cause the puncta formation, but you can also see that in the CF vehicle, the ones that have not been treated at all already have the ORI1 puncta formation, indicating that they are already active even at baseline, which supports the fact that they do have persistent inflammation and supports the fact that ORI1 is an attractive therapeutic for an anti-inflammatory therapy that can be designed. And this just is a graphical representation of the fact that in normal and in CF there is an increase with thapsigargan, but at baseline the CF vehicle is still upregulated. And this was done by a postdoc in the lab, Joe Rennell, that shows that calcium signaling functionally is upregulated in CF neutrophils. So this is the same calcium add-back or calcium switch experiment I showed at the beginning introductory slides, where you start with an extracellular environment free of calcium, and then you deplete the extracellular stores of thapsigargan. And then when you add back calcium, you can see that the CF neutrophils have a higher calcium signal than the normal neutrophils. And again, all of these CF patients are on a CFTR modulator or potentiator, which means that they still have upregulated inflammation and still need to be able to have anti-inflammatory therapeutics to fully be able to benefit them. So in conclusion, we posit that calcium signaling in CF patients is dysregulated despite CFTR modulator use. So as you can see here, you have a relatively normal amount of ORI1 with calcium going in, but then when you have in CF, you have a lot more ORI1 that is activated, which increases inflammation. Because of this, we believe that ORI1 is an attractive therapeutic target, and in fact even that perhaps ORI1 puncta formation could be a novel biomarker of CF inflammation if you're able to get any lung tissue biopsies or even just blood samples from these patients. So with that, I would like to thank the Terran Lab for contributing to my education and teaching me all these really awesome microscopy techniques, as well as the UNC Mars School Lung Institute, who was able to provide all of these lung tissue sections, the histology core, who helped me with the antigen retrieval, the UNC Mars School of Lung Institute Molecular Biology Core, particularly Rodney Gilmore, who was the one who did the actual RNA-scope staining, and Hong Dang, who helped with assistance with data mining from LungMap. And real quick, the Terran Lab is looking for postdocs, and if anybody's interested, you can contact Rob Terran and put in an application. And with that, any questions? It does actually, and in fact, our postdoc, Joe Rennell, has done staining for NFAT, phosphorylation and translocation to the nucleus, and we have seen that there is increased numbers of that as well.
Thank you for sharing your data. I'm wondering if you could go back to some of your slides, especially the one when you showed the bump type and the other pictures. So for the ORI data, it looks to me like the data in the CF is almost like trimodal, right?
Yes.
And actually perhaps in the normal as well. So do you think you have maybe potential subsets of cells depending on the actual amount of content?
Yes, so the question was the fact that there is a very big range of ORI1 punctive formation in these cells, in the normal and in the CF, and the fact that there could be like different populations that are or are not activated. And I do think that that is very, very possible and very true. Even when we're running calcium assays, we don't always have all the cells respond the same or even at all. And I do think that calcium signaling is a very dynamic process, and in particular, these lung macrophages are in paraffin samples with recruited macrophages into the lungs. So I do think it's possible that some of them have been activated, some of them are not, and in particular, like in the normal lung tissue sections, the macrophages are there in the lung lumen, but they're not necessarily responding to any particular infection. So I think that it's definitely like, just like you have patients that are responding to modulator use differently, I think there will definitely be differences in calcium signaling, just patient-to-patient variation.
Yes, if I may follow up on the last data slide as well.
Oh.
On the tracing the blood.
Oh, okay.
Yeah. So was that responsive to ORI or STIM, modulating drugs? The change that you see in the CF?
Yes, so in fact, our lab is developing an anti-ORI1 peptide, and we have tested these in both normal and CF patients, and they respond in both.
So that delta can actually
Minimize.
completely abrogated or just?
We have seen up to 50%. Yeah, so the question is that, in fact, in this particular graph, you can see a huge difference in the calcium signal when you add back calcium, but there doesn't appear to be a difference here. But if you look at just that particular part of it, like, because this magnitude is like up to 5, but this magnitude is probably only 1.5, the difference is lost when you put the graph together. But in fact, when you look at it, just that part where there's the endoplasmic reticulum released calcium, there is actually a difference. So when we're, oh, repeat the question. Oh, I'm sorry. So she was talking about how in the staining for the immune cells, you can see that when it's activated, like traditionally, it's a plasma membrane calcium channel, so you would expect for it to be on the plasma membrane, but you can see in these images, for example, that they're not necessarily what you would consider to be on the plasma membrane. But when I was looking at these lung tissue sections, I want to point out that they are, in fact, sections that have been paraffinized and sectioned for me and deparaffinized. So I do not have much how they have been set in that block. So it all would also depend on what Z position I have imaged them in. So you can image where it's just, you can see that plasma membrane, or you can image at the top or the bottom of the cells too, where you would see that the plasma membrane is like covering the top because you're imaging the top of the cell or the bottom of the cell. And being able to find the plasma membrane as you would think of it being around the cell is much more easier in immortalized cell lines and epithelia and hex cells, whereas in these macrophages, when I was imaging them, I actually wasn't able to find that that specific Z position where they were perfectly surrounding the plasma membrane. So I don't think that this necessarily means that they're not on the plasma membrane. I think that there's some that aren't because they're being produced continuously, but I do think that what you're talking about is more a product of the Z position that this was imaged in rather than them not being at the plasma membrane.
All right, we have a question in the chat, which is, would pharmacological modulation of calcium signaling have adverse effects for lung defense?
That is a very good question. So there have been previous drugs that have been developed, most recently by Exora, that have been a systemic ORI1 modulator, and systemic ORI1 inhibition is in fact immunosuppressive, which is not what we want, especially in these patients that are unable to clear infections. However, when we have been testing our specific peptide, we found that it largely stays in the lung lumen when we have it inhaled or intratracheally deposited in the lungs. And so we have actually found that with our peptide, we are able to help clear infections without having any immunosuppressive effects or being detrimental to survival. And I'm sure that anybody in our lab, including Rob, would be really happy to talk about more about this peptide with you guys.
All right, any final questions from the audience? Great, you have one?
Can I just follow up on that? I mean, I should probably remember this, but I don't. So during T cell activation and the formation of the immunological synapse, is this same ORI1 STIM1 at play?
Yes.
And so if so, I mean, would you potentially be modulating T cell activation, which sort of gets to the last question?
Yes, so we've mostly been focusing on neutrophils and macrophages. However, we have shown that when I've isolated T cells before, that there is an effect on T cells as well, which is to be expected because they are typically the immune cell you think of when you think of ORI1.
All right, please join me in thanking Sasha. All right, please welcome our final speaker of the session, Nam Soo Ju. There we go.
Oh, finally, I can breathe. Thank you for the invitation, Dan and Courtney, and thank you for coming to this workshop. This is my title, and nothing to disclose. And I would like to acknowledge first people who makes this investigation forward, especially Jeff Wine, Carlos Mia, and the WDP, as well as John Engelhardt, who provide precious CF tissue to us. Mucociliary clearance is a vital airway defense system, and it is impaired in CF airways and contribute to lung disease. And CFTR-modulators improve mucus clearance in most CF patients, but people whose mutations are not targeted by these CFTR-modulators remain at high risk. So synergistically increased mucociliary clearance could help such a patient. The question was whether mucociliary clearance impairment found in CF patient is primary or secondary to inflammation infection. The answer came from data by newborn CF piglet and newborn CF ferret, as well as young CF rats with the mucociliary clearance experiment. All the data indicated that mucociliary clearance impairments found in CF patients precede infection. So our main hypothesis is low dose cholinergic plus beta adrenergic agonist, C plus beta, accelerate mucociliary clearance velocity more than their predicted additive effect. So it's synergistic. To do that, we developed a simple but elegant method using ex vivo animal trachea, spearheaded by our colleague Jin-Hyuk Jung. Briefly, we isolate whole piglet or ferret trachea and cut open, mount on prior silicate platform using insect pins, and we drop Xerox toner particles at distal trachea, and the movement of particles from caudal to rostral, so mucociliary clearance, are being recorded using time-lapse digital camera. This is time courses of mucociliary clearance velocity measured in wild-type ferret trachea. So if you use cyclic AMP agonist that's shown in a blue circle and low dose of carbachol cholinergic drug shown in red rectangles, as you can see, they induce mucociliary clearance velocity. And if you combine those two, this is around additive effect. But if you combine those two drugs, and it generates much larger than additive effect, so it's a synergistic effect. So importantly, the synergistic mucociliary clearance velocity can be produced in CF airways, as shown here. Forskolin by itself has almost no increase in mucociliary clearance velocity, and carbachol, low dose carbachol, induce some, and if you combine those two, it produces synergistic mucociliary clearance. The synergy response is statistically significantly larger than arithmetic sum of MCCB induced by forskolin plus carbachol. And this was not species specific because it occurs in pig airway as well. We use, in this case, clinically approved drug, inhalable drug, formoterol as a beta agonist and methacholine as cholinergic drug. As you can see, they produce synergistic mucociliary clearance. As most of the audience well know, the cholinergic drug can induce airway contraction, smooth muscle contraction. That is really a concern to us as well. So to address the question whether cyclic AMP agonists can prevent cholinergic drug-induced smooth muscle contraction, we developed simple method to measure airway narrowing. So sliced like 1.5 millimeter depths of tracheal rings from newborn piglet or ferret was incubated with a cholinergic alone or plus beta adrenergic, and we measure lumen area, and by doing that, we can assess airway narrowing. Quantitatively, it shows here in wild-type ferret trachea, if you use cholinergic drug in the absence of cyclic AMP agonist, you can see the airway lumen area will reduce. That means it's a narrowing with the time. It sustained at least 60 minutes with low dose of carbachol. But in the presence of cyclic AMP, it can prevent cholinergic drug-induced airway narrowing. And David Stoltz and his colleague published a paper demonstrated that a CFTR is hypersensitive to cholinergic agent in terms of airway smooth muscle contraction. So we test whether cyclic AMP can prevent airway contraction in CF airway. And the answer is yes, it does. As you can see, carbachol itself causes airway narrowing, but the presence of cyclic AMP prevents carbachol-induced smooth muscle contraction. So we also check whether ENaC activity is involved in synergistic mucociliary clearance. So in this case, this is also time course of MCCV in the presence of benzamide. Benzamide is inhibitor of ENaC. In the presence or absence of benzamide pretreatment, as you can see, green line, it shows mucociliary clearance velocity significantly increased in, when compared by control, like 6 times in baseline MCCV. However, if we treat with synergy agonist, they produce very comparable mucociliary clearance velocity. That suggests synergy agonists include suppressing ENaC activity. And we also measure whether bicarbonate movement is involved in synergy-driven mucociliary clearance. If you look at using pH stat, as you can see, bicarbonate output here as a rate, and in the presence of synergy drugs, it significantly increased bicarbonate output. It's like around 80%, as shown here. So the second hypothesis is, do the synergy agonists have additional improvement on CF airways that have been treated with CFTR-modulators? To assess that, we developed a multiple particle movement assay. This is showing in left panel ARI culture of human control tissue, human control primary nasal cell, and the right panel is from delta F508. You can see the particle movement around doesn't work. Come on. Yeah. You can see the movement of particle. This is around 5 seconds. And control cell, this is a CF cell. The distance traveled by these particles are much shorter than that of control, as you can see. So combined is expressed as effective diffusivity. You can see this is control, not control, this is CF without any drug. And if we add Trikafta, it increases particle movement. And if we treat with a synergy drug, it can further increase. And if you combine those two drugs, better. So this data actually came from last week from our colleague, Jay and Huang in University of Saskatchewan in Canada, using phase contrast imaging method through a synchrotron X-ray and measure ASL height. As you can see here, these are 4 different peaks and 2 different peak control as a control. The dark black and gray rectangle represent DMSO treatment, and the rest of it is control drug treatment one. As you can see, formoterol, beta-adrenergic drug, increase ASL height, as well as formoterol plus methacholine synergy drug even increase higher. If you combine those, you can see like this, DMSO almost no decrease in ASL height, but if you treat with the formoterol, it increases this. And if you do it formoterol plus methacholine, it appears to be accelerate the rate of ASL height increase. If you apply it different way, it shows synergy agent significantly increase the rate of ASL height increase. So this is our proposal mechanism. So ASL height maintained anion-driven fluid secretion and sodium-driven fluid absorption, and it generates MCCB. And if there is a beta agonist around, it stimulates anion secretion and accordingly fluid secretion as well. So ASL height is increased as well as MCCB. If cholinergic drug is present, it activates CACC and accordingly fluid is increased, fluid level is increased, ASL level is increased. However, it also suppresses inactivity. Accordingly, MCCB is increased as well. So if you combine those two, you can get synergistic MCCB. Summary, beta-adrenergic plus low-dose cholinergic agonist synergistically increase MCCB in ferret, pigs, and most importantly in CF ferret without inducing airway narrowing. Now, our data suggests that involvement of CFTR, TMEM16A, and bicarbonate secretion in synergistic mucociliary clearance. And the synergy agent provided additional improvement with CFTR-modulator. And in future, the key is whether it works in vivo. That will be done most likely this month and next month. And we are also doing a safety and tolerability test in human at Stanford Hospital in the guidance of Dr. Carlos Mia. And these are, we are also planning to do in future. This is it. Thank you. Okay. With granular secretion. Granular secretion, if you combine those two, it synergistically increases granular secretion comparing to single drug. When you combine those two, it synergistically increases granular secretion. We published it. I don't have time to show it, but they do. Yeah, they did. We never tried, right, Jeff? Yeah, we never tried. Yeah, that's a good idea, yeah. Thank you. Yes? Mm-hmm. Yes, that's a good point. Oh, the ASL height, I just show the other slide is close to 200 micrometer. That's, if you measure it in cell culture, typically what you get is 10 to 15 micrometer. But if you measure in either in vivo live tissue or ex vivo tissue, the height of ASL range it like 50 to 300, 400, yeah. That's the measurement by different method actually. One is J use like a synchrotron method, high energy synchrotron. The other is micro OCT like UAB. They use it. When you look at when they use the tissue, the height of ASL is like several hundred as well. Yeah, good point.
So I was just curious, is there any concern about the possibility of altering vascular reactivity? So, I mean, all of these compounds could potentially affect vascular reactivity.
Oh, vascular. Ah, yeah, we didn't think about it. And then this is what we propose is locally inhale. But I don't know if you use it systemically, that may cause a lot of other side effects. But if you use inhalable way, then maybe minimize the.
The only reason I asked is because you had looked at the smooth muscle reactivity,
Right.
And which sort of suggested you might be concerned that it was gaining access to something below the epithelium. So that's why I asked about the vascular.
Oh, because like a Cornell's drug that has been used to test airway sensitivity by inhale way. So people inhale methacholine and whether they are hyperactive to cholinergic drug or not. Yeah, they can access to the receptor supposed to be in the other side, I think. Yeah.
Do we have any more questions from the audience?
Yes, sir. Yeah, go ahead. Oh, well, I don't know whether I correctly understand your question. Why University of Iowa's the oh, because right. I think if I understand correctly, if they use it as like a Teflon disc in live animal, it is a unidirectional way in whole lung. But if they use ex vivo tissue, they use, unlikely us, I always use the way I cut in dorsal central line, that is the heavy muscle area, they cut it and mount it. But University of Iowa, what they use was, that portion is to the center. So when you apply like particle or whatever they use, or fluorescence dye, then it goes all this different way, not like unidirectional center. So that's one of the reasons, I guess. The way they prepare their preparation. That's a good question. I'm not sure whether there is a difference or not. Teflon is pretty big one, but it's interesting. They still can move. When you look at the CT scan, they still can move, but your agar speed is stuck to the ASL. So that's, well, I don't know whether that's a property of the particle you're applying to the system or something else. I'm not sure, yeah, but that's a good question, yeah.
All right, please join me in thanking non-CFTR.
Thank you.
All right, well, thank you very much for attending this session, and I'm guessing that you also took away that there's still There's some great work being done right now on these other therapies, and still a lot to do. All right, enjoy the rest of your day.